So last time we finished up ideas of molecular motors and the ideas of the substrates along which those molecular motors moved. Today we're going to move on and talk about the molecule which we've alluded to several times in here, but really lies at the heart of biology on the planet. And that molecule is DNA, deoxyribonucleic acid. And what we're going to do is we're going to lay out the structure of DNA and talk about its replication. For DNA to be the molecule of life, one of the characteristics it must have is it must be able to replicate itself, because after all, that's what animals and plants do. They replicate themselves. And what's more is there has to be change in that structure over time, because it's the change in the characteristics of the DNA over time which gives rise to evolution. So to be the fundamental life-giving molecule on Earth, these molecules have to pertain to those two major ideas. They need to be able to replicate themselves, and they need to be able to change over time. And we'll see in our discussions today that that's true of this molecule, deoxyribonucleic acid. We're going to see that the components of deoxyribonucleic acid are the 5-carbon sugar, deoxyribose, and 4 bases. The 4 bases are adenine, guanine, cytosine, and thymine. The bases adenine and guanine are said to be purines. The bases cytosine and thymine are pyrimidines. And what we're going to do is we're going to lay out these components, the bases and the 5-carbon sugar of deoxyribose, into such a structure that we're going to generate a molecule of DNA and take that information of the structure of the DNA and extend it into how those molecules can replicate themselves. What we're going to emphasize first, though, is a little bit of chemistry. We're going to emphasize the importance of covalent bonds that are between the individual deoxyribose sugars. Another way of saying that is to say that there are covalent bonds called phosphodiester bonds that link each deoxyribose sugar to its neighbor. So the covalent bonds will be between sugars. Those are the stronger of the bonds. But we'll also find that these bases, adenine, guanine, cytosine, and thymine, have charge on them in such a way that they can interact one with another. Adenine always interacting with thymine. Cytosine always interacting with guanine. And that interaction is through hydrogen bonding. So the bases are going to be hydrogen bonded in the middle of the molecule, where the two sides of the molecules will be held together by the phosphodiester linkage, which are covalent bonds. This will bring us to the idea that a DNA molecule is composed of 2 strands, each strand linked through the phosphodiester linkages of between deoxyriboses, and each one of those deoxyriboses containing a base that can interact through hydrogen bonding with another base. But the 2 strands are oriented differently. What we'll do in walking through the components is we'll see that the organization of the 5-carbon sugar deoxyribose gives the molecule an orientation. The 5-carbon sugar will number the carbons 1 through 5. Where the number 1 carbon will always be covalent linked to a base, and the number 3 carbon will be linked to the number 5 carbon on an adjacent deoxyribose molecule. But that organization gives an endedness to the 2 strands. And we'll see that the 2 strands of DNA are laid out in what is known as an anti-parallel organization, such that one strand is oriented 3' to 5' based on the structure of its sugars, and the other is oriented 5' to 3', again based on the structure of the sugars. Once we've laid the molecule out in its 2 anti-parallel strands, we'll then begin thinking about how we can use that organization to replicate the molecule. If you put your phone away, I'd appreciate it. The replication is said to be semi-conservative. What this means simply is that there's a template strand from which we synthesize a new strand. So every time we replicate a molecule of DNA, we're left with an old strand and a new strand comprising the new DNA molecule. And we'll walk our way through that. For this old strand to provide a template for the synthesis of a new strand, we first have to unwind the molecule. The unwinding of the molecule, because it's going to be thrown into this complex helix, it's thrown into this double spiral helix. The two strands are organized in a helix. So the first thing we have to do is expose the bases by unwinding the DNA and breaking the hydrogen bonds with this molecule called a helicase. The helicase has two functions. As the name implies, it unwinds the helix. But the other thing it does is it pulls apart the hydrogen bonds. And by pulling apart the hydrogen bonds, it's now going to expose those hydrogen bonds of the bases in such a way that the next enzyme, called a DNA polymerase, can come on and begin synthesizing a new strand. Now, if you get down into the details of this, you'll find there are many, many more proteins that are involved in this process. There are a whole bevy of proteins. The only two that I want you to hold on to for here are the helicase that breaks the hydrogen bonds and the DNA polymerase that will form new hydrogen bonds, always adenine to thymine, cytosine to guanine, in addition to forming the phosphodiester linkages, creating a new backbone for the molecule. Now, the enzyme that's forming the new strand, the DNA polymerase, has some rules to it. The enzyme, the DNA polymerase that forms the new strand, always reads the substrate strand. It reads the template strand 3' to 5'. And it can walk along that 3' to 5' end of the molecule continuously all along the chromosome, theoretically. It doesn't do that, but it could. That opens a problem, though, because the DNA polymerase only reads in one direction, the other strand is oriented in the opposite way. That means we have to split the two molecules apart, apply the DNA polymerase, and read back in the opposite direction. This gives rise to something known as Okazaki fragments because we're reading it in fits and starts, and we'll talk a little bit about that. If there's time, we'll talk about the related nucleic acid RNA. The main difference here is that rather than using the 5-carbon sugar deoxyribose, we're going to modify this just a bit. We're going to take a hydroxyl, we're going to add a hydroxyl to the number 2 carbon to make ribose. And the other thing we're going to do is we're going to use a slightly different base. We're going to use adenine and guanine, both purines. We're going to use cytosine we're going to use adenine and guanine, both purines. And the two pyrimidines now will be cytosine, same as in DNA, but we'll change the thymine to uracil. But uracil and thymine are remarkably similar molecules. Thymine is nothing more than 5-methyluracil. So the only distinction between a thymine and a uracil is this methyl group on the uracil to make thymine. But let's start here with DNA structure. And we'll spell this out to say that DNA contains the 5-carbon sugar deoxyribose. The second major idea here is that the deoxyribose sugars are polymerized by covalent bonds called phosphodiester bonds. The third major point of the DNA structure is that it's composed of 2 strands of DNA. The fourth point is that each strand has or shows a, quote, endedness as 3' or 5' based, let me say depending on the deoxyribose orientation. And finally, the two strands are oriented in opposite directions. Well, let's start here simply by describing the structure of the 5-carbon sugar. Deoxyribose is a 5-carbon sugar. Its structure is fairly simple. It's basically a pentagon. We can number the carbons, 1, 2, 3, 4, but we said it was 5 carbons. So there's a 5th carbon up here, sort of as a flagpole. The number 2 carbon has covalently linked to it 2 hydrogens. We'll see later that ribose is distinct from deoxyribose simply by having a hydroxyl at this point. This has implications for the structure of the molecules. But for now, recognize deoxyribose lacks the oxygen on the number 2 carbon. The number 3 carbon, again, has a hydroxyl and a hydrogen. And the number 4 carbon has a hydrogen. And the number 5 carbon has a hydroxyl and a hydrogen, plus a hydrogen. So what you need to recognize from this is it's a 5-carbon sugar. And that the 5-carbon sugar of deoxyribose lacks the hydroxyl at the number 2 carbon. That's what makes this deoxyribose. But to incorporate the deoxyribose into DNA, this number 1 carbon needs to have covalently linked to it a base. For the chemists among you, this is said to be a glycosidic bond. And the glycosidic bond will link this number 1 carbon to a nitrogen on the nitrogenous base. So that's fundamentally the structure of the ribose. But in addition, the other point I want to make up here is that this number 5 carbon can have on it an oxygen, a phosphorus, an oxygen, a phosphorus, an oxygen, and I wish I had more room, another phosphorus. This is a triphosphate. If you go through this, and I won't go through all the details, there's a double-bonded oxygen and an O negative down here. This phosphorus has a hydroxyl on it. But this is said to be the deoxyribose form of a base trinucleotide. So that if this base out here was an adenine, this would be a trinucleotide. It does have 3 phosphates on it, so it's an adenine trinucleotide. It could be a cytosine trinucleotide. It could be a guanine trinucleotide. It could be any of the bases. It could be a thymine trinucleotide. Now, if this were not deoxyribose, but rather were its related cousin, I'll put it up here, ribose. Ribose would have a base here. It would have a hydroxyl at the number 2 carbon. It would have a hydroxyl here, a hydrogen, a hydrogen, a hydrogen. The number 5 carbon would have an hydroxyl and a hydrogen and 2 hydrogens. So the only distinction here between this structure and this structure is the ribose here has a hydroxyl. But if we were to put an adenine out here and now modify the number 5 carbon, so this is an O, P, O, P, O, P. Count the phosphoruses, 1, 2, 3. If this were an adenine on a ribose, this would be adenine triphosphate. So this is true. This is the ATP we've been talking about. This is the ATP that's used as the energy source to drive the motor molecules. This 5-carbon sugar of ribose can have any of the 4 bases attached to it. It could be guanine or cytosine or uracil or adenine. The only distinction back here is that we can make the same molecule. We can add a triphosphate out here having a base at the number 1 carbon. Could be adenine, thymine, cytosine, or guanine. This base is linked to the number 1 carbon and has a triphosphate, but the only difference is this structure is based on ribose. This structure is based on deoxyribose. But they're fundamentally the same thing. We're now going to use this triphosphate to begin assembling the DNA molecule. So let me start with a clean slate. So let's say, over here, we have our 5-carbon sugar. 1, 2, 3, 4, and 5. It's going to be linked at the number 5 carbon with an oxygen, a phosphorus, an oxygen, a phosphorus, an oxygen, a phosphorus, and an OH. That's the triphosphate. Now, we could have any one of the 4 bases, adenine, thymine, cytosine, or guanine, attached to the number 1 carbon by a covalent bond. I'm showing that simply as a straight line out to the base. That's a covalent linkage between the number 1 carbon and the base. Now, the next character I'm going to bring in here is another deoxyribose molecule. Again, with 1, 2, 3, 4, and 5. Now, this number 3 carbon on the first one has a hydroxyl. And just as we showed up here, we're going to bring in this deoxyribose linked to a phosphorus, an oxygen, a phosphorus, an oxygen, and a phosphorus. And I won't put all the other decoration on it. Right now, I'm going to do this by waving my hands. Here a little later, we're going to show how an enzyme does this trick. But the enzyme is going to be something known as DNA polymerase. And what the DNA polymerase will do is it will come in and it will recognize the triphosphate on this number 5 carbon. And it will recognize the hydroxyl on the number 3 carbon. And what it's going to do is it's going to take off this hydrogen, and it's going to take off the hydroxyl. Nope, I'm sorry. It's going to take off the hydrogen from this hydroxyl. It's going to take a hydroxyl from this to make water. And the end result is going to be this number 5 carbon linked to this phosphorus. Sorry, it's number 5 carbon linked to an oxygen, linked to a phosphorus, linked to the oxygen. So now we have a bond that goes from the number 5 carbon to an oxygen, to a phosphorus, to an oxygen on the number 3 carbon. This is said to be a phosphodiester bond. It's this phosphodiester bond that provides the covalent linkage that ties this deoxyribose to this deoxyribose. This number 1 carbon will have a base on it. It could be any of 4. We can repeat this pattern again. So that again, here's another deoxyribose 1, 2, 3, 4, the number 5 carbon. It will come in with a phosphate. It will come in with a phosphorus, oxygen, phosphorus, oxygen, phosphorus, OH. But the end result is going to take this hydroxyl on number 3 carbon and link it through its oxygen to a phosphorus to the oxygen on this carbon. So now we have again a carbon, oxygen, phosphorus, oxygen, carbon bond, the phosphodiester linkage. So now we have formed a covalent bond between the second and the third deoxyribose. And we can repeat this over and over and over again. So here again, I won't go through all of the painful parts of it, but the number 5 carbon here will be linked by an oxygen to a phosphorus to an oxygen bond to this number 3 carbon. We can do this millions of times, and an individual DNA molecule, an individual chromosome will have maybe a million of these strung one to the next, all linked by these phosphodiesterone linkages. The other point I want to make here is that this is the number 5 carbon. So this is said to be the 5' end. This is said to be the 3' end. It doesn't matter whether we have 4 bases or 4 million, it will be always oriented with a 5' and a 3' end. That's for this strand. But you remember from the introduction, we said that one of the hallmarks of DNA is that the 2 strands are oriented in opposite directions. So now what I'll try to do is draw the other strand, and I always struggle with this because it's upside down, but bear with me. So there's the number 1 carbon. So there's the oxygen, number 1 carbon, number 2 carbon, number 3 carbon, number 4 carbon, and number 5 carbon. And there's the oxygen between them. As before, this number 1 carbon is going to carry a base. And I'll mention it here, we'll emphasize it in a moment. These bases here are going to be held together by hydrogen. Bonding. But this deoxyribose, and again, I'll make sure we have the 2 hydrogens on it to emphasize the point this is deoxyribose. But it will have on this number 5 carbon an O, P, O, P, O, P, OH. Then the next carbon will come in, the next structure will come in. It now has the oxygen, number 1 carbon. Let me bring it a little higher, farther away. 1, 2, 3, 4, and 5. This number 3 carbon has an OH on it. The number 1 carbon again has a base. This base will hydrogen bond to this base. We could put the proper names in here if you'd like. If this is an adenine, this has to be a thymine. If this is a cytosine, this has to be a guanine. But now we're again going to use the hydroxyl on number 3 carbon. And again, just as we showed on the other side, we're now going to form this O-P-O bond, a phosphodiester bond linking the number 5 carbon to the number 3 carbon. We can continue this story on with the number 1 carbon here, the bases, and a base, and an O-P-O linkage to the number 5 carbon. Now you note what I've done. I'll put one more in just to make it complete. It's 1, 2, 3, 4, and 5. So the number 1 carbons have the bases. They are going to base pair. They are going to base pair by hydrogen bonding. The number 3 carbon is linked by an oxygen to a phosphorus to an oxygen bond to this number 5 carbon. So what have we done? What we have done is we've laid out this strand in such a way that this is the 5' end and this is the 3' end. But we've organized the other strand in such a way that this is the 3' end and this is the 5' end. This is said to be the anti-parallel arrangement of DNA. So if you look at this, you can recognize that the two strands are pointed in opposite directions. The sugar molecules are all held together by covalent bonds in the form of these phosphodiester linkages. But the two strands now are being held together by the much weaker hydrogen bonds between adenine, thymine, and cytosine and guanine. This is a fundamental idea of the organization of the DNA molecule. And it was Watson and Crick who recognized that this had to be the organization. It had to be the bases pointing inward and the phosphates pointing outward. In the early 50s, when they were looking at this and trying to sort out the molecule, all they knew was that Chargaff had isolated DNA, and he had broken it down to his component parts. And Chargaff had shown that there were equal amounts of adenine and thymine and equal amounts of cytosine and guanine, suggesting that those two must bind together. Then Rosalind Franklin, working with a guy named Wilkerson, had done some X-ray diffraction data, had gathered X-ray diffraction data on crystallized DNA. And when they looked at that, they saw a repeating pattern. And they saw the repeating pattern at a regular interval. And what they were looking at was the interval of the bases and the sugars. But what they couldn't sort out, and what Rosalind Franklin was unable to sort out, was whether the bases were pointing inward or pointing outward. And when Watson and Crick looked at the X-ray diffraction data, they said the bases have got to be pointing inward. They must be pointing inward based on X-ray diffraction data. And to show that, what they did is they went to the machine shop at Cambridge, and they had the machinists at Cambridge form scale models of adenine and thymine. So they had these scale models of the bases, and they would sit down at a table and try to fit them together mechanically. And they found the only way they could fit them together was adenine had to work with thymine, cytosine had to work with guanine. It was the only way they could fit mechanically. And then once they realized that these fit mechanically together in the middle, then they began to recognize that the helical structure, which they saw in X-ray diffraction, resulted mostly from the fact that these bases, once they were base paired, didn't have much charge on them. And because once they were base paired and they didn't have charge, they were hydrophobic. And because they were hydrophobic, they would point inward and exclude water. And the most efficient way to exclude the water from these hydrophobic regions was to twist it into a helix. The other forces that are acting here is that these bases, and I'll show you a picture of this in a moment, these bases are flat, plate-like structures. And because they're flat, plate-like structures, they'll stack one on top of another. And it's that stacking energy in van der Waals forces, which then allows for this thing to throw itself into the helix. So it's both the effects of water and the stacking forces of the plate-like structures of the bases, which throws this molecule into a helix. Yes?
Can you explain where the rest of the triphosphate group goes?
So where does the rest of the triphosphate group go? Well, these two go out as a diphosphate. And some of these diphosphates then are going to go back into the mitochondria. And the diphosphates then will be linked with a monophosphate, or maybe sometimes they'll be broken down into individual phosphates and reused to make ATP, for example. So the phosphates are basically recycled. But the highly energetic part of the molecule is when it has the triphosphate, as with ATP. That's a triphosphate. So you store a lot of energy in that triphosphate structure. And that energy stored in the triphosphate structure, just as we saw the other day with converting chemical to mechanical movement, chemical energy to mechanical movement, that energy is stored here. That energy in the triphosphate then can be used by the enzyme to form this phosphodiester linkage. Yes? endedness. And what I mean by the endedness is what we're saying here. We have a 5' end and a 3' end and a 3' end and a 5' end. So let me make that an E. Yes?
So what started as a OH bonds serve as basically like the H will fall off and phosphate can come in there, like the phosphate groups will come in there.
So we use the hydroxyl to form the hydroxyl and the number 3 carbon. We use that to form the phosphodiester linkage. I think we keep this oxygen. If an organic chemist would come up here and tell me differently, I'd have to agree. But I think we keep this oxygen, and then the phosphate, I think this oxygen then replaces the one on this carbon, I think. But we might lose the hydroxyls on both of these, I'm not sure. Yes?
So the phosphodiester bond is between the phosphorus, oxygen, and phosphorus?
So the question is, what do you mean by a phosphodiester bond? The oxygen phosphorus oxygen bond is said to be a phosphodiester bond. And
All three of those atoms.
it includes all 3 of those atoms. And that's the phosphodiester bond. And we're going to see this is the way we link together these sugars. And we get to ribose. We're also going to link these together in great long chains too. It's just the only difference will be the hydrogen here and the difference in the bases. Yes?
Where's the loss of water coming from?
It's from the hydrogen here, and I think it's coming, I think there's a hydroxyl out here someplace. Maybe it's from the 1 hydroxyl from this number 3 and a hydrogen from the number 5. I'm not sure, but that's where it's coming. It's coming from one of those. Yes?
So the oxygen and the phosphorus from the phosphodiester bond, that's part of the trinucleotide.
Okay, so this is a question that came up last time too, and I'll spell it out. This is a term of nomenclature. And when we have the 5 carbon sugar, whether it's deoxyribose or ribose, if we have the 5 carbon sugar linked to a base, then out here in the number 5 carbon we have the OPO 3 phosphates. When we have the triphosphate plus the base, the terminology here is a nucleotide. If we have the 5 carbon sugar without the triphosphate, so this is a carbon, hydrogen, or hydroxyl hydrogen, this is said to be a nucleoside. So the side is the sugar plus the base. The tide is the sugar, the base, and the triphosphate. And I should say, too, that these bases all have their independent names, and they can appear in biology independently as adenine, guanine, cytosine, and thymine, which, by the way, is 5-methyl uracil. Does that answer your question? Yes?
Is there maybe much other difference between a nucleotide and nucleoside, like the nomenclature specifically, or is that not super relevant?
So the question here is, what's the difference and what's relevant? Well, if you're a chemist, it seems to be really relevant. So in the nomenclature, if we want to talk about nucleotides, then we know, then we say nucleotide. Then we know it has 3 phosphates. If it's a nucleoside, we know it lacks the phosphate and it's just with the sugar and the base. And then there are the names of the bases. That's just to keep the nomenclature straight. And if you read some of this in molecular biology, they'll use these terms without defining them particularly. And so you just need to know the language. And I only bring it up here because the question was asked, what's the difference among them? Anything else? Okay. So what we've done here then is we've laid out these 2 molecules. We have shown that the sugars are linked by phosphodester bonds, which are covalent bonds, and the bases are linked together here by hydrogen bonds. Let me take a minute and show you a couple of slides to kind of round that out. So the first slide is just a pretty picture of what we had on the board. And it shows all of the relevant characters. So here's the 5-carbon sugar. They have the hydrogen in red to demonstrate that this is deoxyribose. So if you count the sugars 1, 2, 3, 4, and then 5, the number 1 carbon they're showing here, covalently linked by the glycosidic bond to the guanine. Here it's the cytosine. Here's the thymine. Here's the adenine. Those are the bases. But what mostly this is showing is this carbon, oxygen, phosphorus, oxygen bond between the number 5 carbon and the number 3 carbon. That's the phosphodiester linkage. The other thing it's showing is that this is the 5' end of this strand, and this is the 3' end of this strand. What it's not showing is the other strand, which would be over here someplace, where the bases would be interacting one with the other through hydrogen bonds, and where the number 5 carbon here is at this end of the molecule, the number 3 carbon would be on the corresponding complementary strand. Now, this is a nice figure. Don't memorize it, not for here. But I like this figure because it brings together a number of ideas. It shows out here on this nitrogen of the thymine and on this nitrogen of the adenine, it shows that these are bonded to the sugar. So this would be to the number 1 carbon, the deoxyribose. Same here. That's also true down here with the deoxyribose to guanine and to the pyrimidine cytosine, shown at this nitrogen. Now, when these molecules come close together in the middle of the DNA strand, they hydrogen bond. And the hydrogen bonding here is between this double bonded oxygen with its negative charge and the amine nitrogen, or hydrogen rather, out here on this amine group. And down here, there's a negative charge in the nitrogen and the ring structure of adenine that can hydrogen bond with the positively charged hydrogen on this nitrogen within the thymine ring. So the only way we can bring these molecules together and have them properly bond one with the other through these hydrogen bonds is to have them fit together in such a way that there's hydrogen bonding here between adenine and thymine, and in this case, between cytosine and guanine. Now you'll notice here that this bonding, CG, has 3 hydrogen bonds. One here between this positively charged hydrogen, the double bonded oxygen, again, hydrogen to the negative charge on the ring structure, and the double bonded oxygen to the hydrogen. There are 3 hydrogen bonds here, and in the early days of molecular biology, one of the ways they would think about DNA molecules is they would heat them. And they would heat them and put enough heat into the molecules that they would melt. And when they melted, they would come apart into 2 strands. And they could measure what proportion of the DNA was in 2 strands. If you put your phone away, I'd appreciate it. Then what they found was that in regions where they had lots of cytosine and guanines, it took more temperature to thaw those bonds, to separate those bonds. So that indicated to them the CG bonds were stronger than the AT bonds. And so then from there, they began to sort this out and find out what the hydrogen bonding was that was holding the molecules together. So far, the major ideas are that we have covalent linkages between sugars, we have hydrogen bonding between bases, and that the 2 strands of each DNA molecule are pointed in opposite directions. What I want to do up here is I want to take that information and incorporate it into the fundamental idea of how DNA molecules replicate themselves. One of the insights that Watson and Crick and others had in the early 1950s, late 1940s, was that if you were looking at the molecule which was at the heart of how biology functioned on the planet, that it had to have a couple of major characteristics. And to define life, life must replicate itself. It has to reproduce. And so to be the molecule of life, these molecules needed to have a mechanism of replicating themselves. And that's what we're going to talk about now, is the concept of DNA replication. The characters we're going to involve here in DNA replication are first a molecule, an enzyme called helicase. The function of the helicase is to first unwind the helix, and secondly, to break hydrogen bonds between bases. The other major character here is going to be a molecule called DNA polymerase. And what DNA polymerase does is that it finds the complementary nucleotide and forms phosphodiester bonds between adjacent deoxyribose molecules. The third point I want to make here, leave myself enough room. The third point over here is that DNA polymerase reads DNA from 3' to 5'. So those are the fundamental ideas that we're going to now incorporate into this next step. And I'm going to simplify this a whole lot. I'm going to take the complicated structure I have above with all of those deoxyriboses linked by phosphodiester bonds, and I'm going to replace that by this blue line. And then all of those hydrogen bonds now are going to be simplified in this image so that this red vertical line is meant to be the linkage to the number 1 carbon on the deoxyribose. And then I'm just going to put the bases in. And I'm going to put these in more or less at random. So this is an adenine, thymine, cytosine, guanine, cytosine, adenine, adenine, guanine, cytosine, cytosine, guanine, adenine, thymine, guanine, adenine, cytosine, guanine. Now the complementarity here tells us this adenine has to base pair with its hydrogen bonding to this thymine, a thymine to an adenine, a cytosine to a guanine, and so forth. It's a guanine and a cytosine. I hope I haven't put any mutations in there. Now the next character is now going to be this so-called helicase. And I'm going to show the helicase as basically a kind of wedge. But the helicase has two functions. The helicase is going to take the structure, which I haven't shown here, as a helix, and unwind the helix. So it unwinds the helix and basically flattens it out. And then it inserts itself in between the bases. It basically breaks the hydrogen bonding. These are not exceptionally strong bonds. So it's now going to break the hydrogen bonding. And so it's going to walk down the molecule here as this wedge, separating the two strands. Then as the two strands are separated, the next character in the play comes along. This molecule is said to be DNA polymerase. Now remember, I said the DNA polymerase has to read the strand 3' to 5'. So that means this DNA polymerase is going to have to find its way to the 3' end of the molecule. The DNA polymerase is now going to walk along this strand from the 3' end toward the 5' end. Each time it encounters a base, in this case a cytosine, it's going to plug in a guanine. And then when it finds the next one, in this case is the guanine, so it has to plug in a cytosine. In addition to finding the correct complementary bases, then it will also form the phosphodiester linkage. So it's using the energy in the triphosphate of the incoming nucleotide to form this phosphodiester linkage. And this continues on down the line, where this thiamine base pairs with an adenine, covalently linked then to the backbone. And this will go on, this cytosine to guanine, adenine to thymine. And this will continue in an unbroken fashion, moving from 3' to 5' end. As we generate this strand, two things to note. First, this is said to be the leading strand. And the second is that this leading strand can theoretically be made continuously as the DNA polymerase marches along the molecule 3' to 5'. But now we run into kind of a problem. The polymerase can only read 3' to 5', and yet we said with semi-conservative replication, one strand is a template strand, and the other is a new strand. So again, what we're seeing here is we're using the CGTCA, that's the template strand. And now we're generating the new strand, GCA, GT, and so forth. This is said to be the new strand. Well, up here, this is going to be the template strand. But if this is the 3' end and this is the 5' end, then knowing that we have the antiparallel organization, this is now the 5' end and this is the 3' end. So imagine this thing being several million base pairs long. The helicase comes in and opens it up. It's fine for this strand. We can walk right along the strand and read it 3 to 5. But now what we have to do for this other strand, the other strand which is running 5 to 3, we now have to wait for the helicase to open this thing up. Open it up to such a point that someplace down here, after this has been split apart, after the helicase has pulled the two apart, along comes DNA polymerase. It does the same thing. It will find the cytosine base pair it with a guanine, the cytosine with a guanine, guanine with a cytosine. It will form the appropriate phosphodiester linkages. It's doing the same thing here as we showed down here. But what's happening is this is running 3' to 5'. So this DNA polymerase functions in fits and starts. What we mean by that is that it will find this open 3' end, read it 3 to 5, complete that new strand, and then wait for the 2 strands to be separated. Another DNA polymerase comes on here and reads it back in the other direction, and reads it back in the 3' to 5' direction. So what we're doing here is we're splitting the 2 strands apart. The leading strand can be read straight through, but the lagging strand is read and synthesized in fits and starts. And if you have heard of Okazaki fragments, that's where this comes from. The Okazaki fragments were the fragments they found when they were doing these initial experiments, and we'll pick this idea up tomorrow.